Hello everyone. Let's talk about Ecdysozoans in this second part of the lecture. The species Poor Onychophora form the tiny sister group to the more than 1 million arthropod species with articulated appendages. X unites these under the name Euarthropoda. Within the Euarthropoda, the highest-ranking sister groups are the Chelicerata and the Mandibulata. The division of the appendages into articulated limbs evolved later in the stem lineage of the Euarthropoda and do not, therefore, contribute to the characterization of the Arthropoda, from which the name Arthropoda is derived. I will deal first with several autapomorphies of the Arthropoda before discussing the apomorphic peculiarities of the Euarthropoda. The cuticle, with its antiparallel arrangement of neighboring chitin chains and their covalent connection to protein, is secreted by the ectoderm. Once produced, the cuticle cannot be enlarged. The loss of mobile cilia on the surface of the body is a consequence of the evolution of the hard chitin cuticle. The head of the Arthropoda is formed from the acron and probably the first 3 segments with appendages. The antennae of the Onychophora are homologous with the antennae in the ground pattern of the Euarthropoda. A dorsal pericardial septum spans the body horizontally. Hemolymph circulates in arteries that exit from the heart and in sinuses of the body cavity. Veins are absent. The stem species of the Arthropoda was an aquatic organism. The transformation of the Onychophora to terrestrial living must be interpreted as a case of convergence to the evolution of terrestrial Euarthropoda. In connection with the change to land came the development of tracheae. However, this cannot be homologized with the tracheae of the Euarthropoda. Sickle-shaped blades open in the oral cavity. A large, lensed eye lies dorsally on both of the antennae bases. Homologization with the light sensory organs of other articulata is not possible. Consequently, an independent evolution in the stem lineage of the Onychophora can be hypothesized. Pairs of salivary glands open in the oral cavity. In comparison with the Euarthropoda, the ground pattern of the Onychophora includes a number of plesiomorphies. I now list several characteristic features of the Euarthropoda. Evolution of solid, flexible chitin plates. There is a minimum of a dorsal tergite and a ventral sternite per segment. In connection with the formation of the exoskeleton, the whole body musculature transformed into separate cords with cross-striated musculature. Jointed appendages with 2 branches from the basal unpaired protopodite, an endopodite and an exopodite arise. 1 pair of eyes in the acron, composed of multiple ommatidia. 4 separate ocelli in the middle of the head. Reduction of the segmental excretory organs to 6 pairs on segments 2 to 7. Larva with 3 pairs of appendages. In the arrangement of the head from the stem species of the Euarthropoda, the Chelicerata and the Mandibulata have taken separate paths. In the stem lineage of the Chelicerata, 3 trunk segments were united with the original Euarthropoda head to form a new entity. The antennae of the first segments are reduced. The chelicerae evolved from the appendages of the second segment as 3-part grasping organs. The following 5 appendages became uniramous structures for locomotion. In the stem lineage of the Mandibulata, A subdivision of the head took place into a pregnathal section with differentiation of a second antennae pair, and a gnathal region with evolution of mandibles and 2 pairs of maxillae. With the fusion of 3 trunk segments to the head of the Euarthropoda, a division of the body into 2 large stigmata, prosoma and opistosoma, took place in the stem lineage of the Chelicerata. The prosoma consists of the acron and 7 segments. Subdivision of the opistosoma into the mesosoma of 7 segments with appendages, and the metasoma from 5 metameres without appendages. The lack of the pair of appendages, which are designated antennae 1 in the crustacea, is postulated to be a product of reduction. The reduction of appendages on prosoma segment 1 is correlated to the reduction of the corresponding brain section. In the stem lineage of the Chelicerata, the appendages on prosoma segment 2 evolved into 3-part grasping organs. The 2 distal articles form a pincer-like structure. The primary paired genital openings are shifted from the posterior part of the body to the anterior part of the opistosoma. From the ground pattern of the Euarthropoda, paired lateral compound eyes and 4 median eyes were carried over into the ground pattern of the Chelicerata. The alternative continuance within Chelicerata can be used as the first justification of the systemization into the sister group Pantopoda and Euchelicerata. The original 4 median eyes are only found in the Pantopoda. A reduction to 2 median eyes in the stem lineage of the Euchelicerata must be postulated. The pedipalps of the Arachnida and the parapalps of the Pantopoda are in the same instance comparable filler-like appendages having an identical location behind the chelicerae. The widespread use of the same term implies a homology between the appendages of the Arachnida and the Pantopoda in their construction as filler-like organs. However, in a sister group relationship between the Pantopoda and the Euchelicerata, this is not possible. With a length of up to 3/4 of a meter, the Scyphosaura, the sister group to the Arachnida, are the giants of the recent Chelicerata. Comparable requirements necessary for colonizing the land in the Arachnida and the Tracheata have led to remarkably similar yet independent answers and to homonymous terms. For the systemization of the Arachnida, 2 partly divergent assessments are available. The evolutionary success of the spiders with a genesis of 35,000 species is connected with the evolution of a unique feature in the stem lineage of the Araneae, the ability to spin a web. The 2-part subchelate chelicerae appear in the Araneae in 3 different forms. Orthognathy is illustrated in Figure 7. The basal segments sit horizontally on the prosoma. The terminal segments fold in parallel to one another ventrally. Captured prey is clamped between the basal and terminal segments. Crustacea and Tracheata are sister groups. Crustacea have retained the aquatic habitat of the stem species of Mandibulata. Many features of crustacean organization were carried over from as plesiomorphies from the ground pattern of the Mandibulata, such as the second antennae. However, the nucleus eye, 4 pigmented cup ocelli, have moved together and is a feature that can undoubtedly be interpreted as a derived characteristic of the crustacea. The terminology concerning the division of the crustacean body is not uniform and can lead to misunderstandings. X2000 uses the major terms. Scylla, a Eumalacostracan genus, illustrated in Figure 8, exhibits two autapomorphies of the Eumalacostraca. The exopodite of the second antennae is a large, flattened scale, a steering organ for swimming. The appendage-less abdominal segment fuses with the sixth segment of the pleon. The furca of the telson is reduced. Pleopods 6 are strongly developed. They are directed diagonally backwards next to the tergoplate of the telson, and together with this form the tail fan. In phylogenetic systematics, the most parsimonious assessment of the facts can lead to only one conclusion. In the stem lineage of Tracheata, during the change to land, a tracheal system evolved on the trunk with strongly segmentally arranged pairs of spiracles only once. Whosoever wishes to postulate a multiple independent origin must give reasons for setting aside the principle of parsimony. Synapomorphic congruences do not exist between the Chilopoda and the Insecta. For the three congruences between the Progoniata and Insecta, labium, coxal organs, and style, established objections exist for each against their interpretation as synapomorphies. The absence of median eyes in the Chilopoda and the Progoniata is a derived secondary state in comparison with their existence in the crustacean and Insecta. The lateral ocelli without a crystalline cone in the Chilopoda and the Diplopoda are apomorphic in contrast to the compound eyes of the Crustacea and Insecta. A connection may exist between the cryptic habitat of the Chilopoda and the Progoniata. Myriapoda, which, for reiteration's sake, includes both the Chilopoda and the Progoniata, is characterized by the loss of median eyes and modification of the compound eyes into groups loosely clustered lateral ocelli. A good example of an autapomorphy of the Chilopoda is the maxillipeds. The first trunk segment appendages are transformed into large claws or fans. Each claw contains a poison gland. Paralysis or death of the prey pierced by the fans occurs due to the poison. I follow the common equalization of the names of Insecta and Hexapoda for a taxon comprising all insects. The Insecta comprises 5 monophyla of primary wingless organisms: the Diplura, Protura, Columbula, Acugnata, and Zygentoma, as well as the winged Pterygota. Phylogenetic systematics must reject the traditional division into Apterygota and Pterygota. In the stem lineage of the Insecta, a division of the trunk into a thorax of 3 segments and an abdomen of 11 segments plus telson took place. One pair of antennae was carried over from the ground pattern of the Tracheata into the stem species of the Insecta. Biting, chewing mouth parts are widespread among the Insecta. Mouth parts are modified for piercing in this mosquito of the genus Culex, sucking nectar from flowers in the alfalfa butterfly of the genus Coleus, and sopping up liquids in the housefly Mosca domestica. The 3 pairs of locomotory appendages were taken over from the stem species of the Tracheata into the stem species of the Insecta. They are a plesiomorphy in their ground pattern. Cerci exist in the Diplura, Archaeognatha, Zygentoma, and some basal taxa of the Pterygota on the 11th abdominal segment. 4 frontal ocelli belong as a plesiomorphy in the ground pattern of the insecta. Compound eyes were taken over from the ground pattern of the mandibulata. They form therefore a plesiomorphy of the insecta. Digestion takes place primarily in the stomach or midgut, and excretion takes place through Malpighian tubules, of which there are 6. They occur at relatively few horizons, mainly in lacustrine deposits, but the number of fossils present may be enormous. The finest specimens are found in amber, where preservation may be exquisite. The arrival of the flying insects marks 2 stages in hexapod evolution. They first occur in the Carboniferous. The most primitive forms were those with wings that could not be folded back over the body, and in which the larvae were aquatic. The ability to fold the wings was an asset that enabled them to creep into crevices and retreat from predators. In the Permian, new groups appeared with the larval stage as worm-like groups. Mesozoic fauna consists of dipterans and wasps, demopterans, and other hymenopterans and lepidopterans. The insect fossil record is a great success story. At an early stage, the insects had developed basic adaptations for terrestrial life: efficient air-breathing tracheae, a waterproof cuticle which prevented desiccation, efficient walking legs, and later wings providing mobility. On land, they found and exploited new niches. As plants evolved and diversified, so did insects. Nematodes are bilaterally symmetrical, unsegmented worms that have longitudinal muscle but not muscle in a circle around the body. Aside from many free-living species, some nematodes are parasites of animals and plants. In some parasitic species, females burrow through the intestinal walls of some mammals and release their eggs, which become lodged in the muscles. Eating undercooked meat of such animals can transmit the worms to humans. Euarthropods have a skeleton formed from plates and jointed appendages. The Chelicerata with mouth parts that function as fans or pincers, and the Mandibulata, which is divided up into the Crustacea and the Tracheata, are the highest ranking sister groups within the Euarthropoda. Insecta, the insects, have 3 pairs of locomotory appendages, and Myriapoda with 1 or 2 pairs of appendages on every body segment are sister groups of the Tracheata. That is all for now. Thanks for watching. See you next time.